Hello everyone, I am Himanshi from DNA Education and today we are going to start a new topic, that is life processes. So let us begin with the first topic of the chapter, which is nutrition. As you know that nutrition is the process of taking in food inside the body and then its utilization by the body. So basically these are two aspects of nutrition. One is intake of food from outside into the body and second is the utilization of food by the body. Now there are two major type of nutrition as you all have studied in the lower classes. One is autotrophic nutrition and the other is heterotrophic nutrition. Let us first study about autotrophic nutrition. Autotrophic nutrition, as we know, is the nutrition in which organism makes its own food, and this type of nutrition is mainly seen in green plants because green plants have the ability to make their own food using raw materials from the environment. As we know that photosynthesis is the process by which green plants prepare their own food, and this is the equation representing photosynthesis in which carbon dioxide and water are taken in by the plant body in the presence of sunlight and chlorophyll, and it is converted into glucose, oxygen and water. This glucose represents the food that is prepared by the plants and is stored in their bodies as well as available for other organisms for consumption. Now let us study how plants take in the raw materials, that is how the process of nutrition goes in plants, how they take in the raw materials to make their own food. As we know that water is absorbed by the roots of the plants and then supplied to the leaves through vessels. Similarly, carbon dioxide is taken in by the plants through tiny pores present on the surface of their leaves. Let us first understand the structure of a leaf of a plant. This diagram shows the cross section of the leaf of a plant. Suppose there is a leaf and we cut the leaf cross sectionally and then observe the section, we can see several layers in the leaf. The uppermost layer is the cuticle. It is the protective layer and prevents loss of moisture from the leaf. Below cuticle is the epidermis. This is the first cellular layer of a leaf and it protects the subsequent layers. The leaf on both sides is sandwiched by epidermis. Below epidermis is palisade parenchyma. These are elongated cells of parenchyma and these tiny cells, they represent chloroplasts because chloroplasts are the cells in the leaves which contain chlorophyll, and chlorophyll is very essential for trapping sunlight. Below palisade parenchyma is spongy parenchyma, and in the lower epidermis of the leaf you can see different cell structures. These are the guard cells and there is a pore between the guard cells which is called the stomatal pore. It is this pore in the leaves through which gases come in or goes out. Now let us study the structure of the stomatal pore in detail here in this diagram. This is a standard diagram of stomatal pore. As you can see that these are two kidney shaped cells. These two kidney shaped cells are called guard cells. These are surrounded by several cells which are called subsidiary cells. Now you can clearly see that the outer surface of the guard cells is thin while the inner surfaces are thick. These thick surfaces are bound by a pore. This pore is called the stomatal pore. This is the nucleus and these cells represent the chloroplasts which again contain chlorophyll. So whenever plant absorbs water, the cells swell up. Due to swelling up, the pressure increases and the guard cells open up and the pore opens. Because of this open pore, gases can come in or go out from this cell. Similarly, when the plant loses water, the cell becomes flaccid and the guard cells, as you can say, they become compact, flaccid and the pore closes and the gaseous exchange is stopped. So this is how the stomatal apparatus works in the exchange of carbon dioxide from the atmosphere. Now after studying the autotrophic mode of nutrition, let us move on to the heterotrophic mode of nutrition which is seen in animals. Heterotrophic nutrition can be of several types. For example, in fungi we see saprotrophic mode of nutrition. In this mode of nutrition, The fungi, they digest the food particles outside their body and then absorb the digested food inside their bodies. Another mode is the decomposition mode, which is seen in microbes, and one more important mode is the holozoic mode of nutrition, which is seen in human beings. Holozoic means, holo means complete. So in holozoic mode of nutrition, the entire food particle from outside the body is consumed inside the body, as we do in our nutrition. So let us understand the holozoic nutrition first in amoeba. Amoeba is a unicellular organism and it does not have any definite shape. So it has an irregular body shape, and this is a nucleus and a cytoplasm. Whenever there is a food particle surrounding the cell of amoeba, it projects some projections, finger-like projections. These are called pseudopodia. These pseudopodia, they project and tend to surround the food particle and then completely take in the food particle. This membrane bound food particle is now called food vacuole. The digestion of the food particle takes place inside this food vacuole. Afterwards, the digested food is absorbed by the rest of the cytoplasm, and this is called assimilation. Finally, the waste materials are expelled outside the body of the amoeba through the membrane. So in case of amoeba, we see that a single cell body of amoeba completes the complete process of nutrition, digestion, assimilation and excretion. Now when in case of humans, we see that we have an elaborated system of digestion in which different parts have different functions. Let us study the digestive system in humans in detail. First of all, we have the mouth. The mouth consists of four main parts: the teeth, tongue, lysozyme and saliva. Teeth, as we all know that we have 32 teeth, or we can say there are two jaws in which 16 teeth are aligned in each jaw, and teeth help in chewing and grinding the food and also helps in mixing the food with saliva. Next is the tongue. Tongue is a muscular organ and it again helps in mixing the food with saliva, and it also helps in engulfing the food into the food pipe. Next is the lysozyme. Lysozyme are small enzymes present in the watery fluid of the mouth, and they help in killing the bacteria and germs. Saliva. Saliva is the primary liquid which is secreted by salivary glands inside our mouth, and this saliva contains salivary amylase. Salivary amylase is an enzyme. Whenever we eat food in our mouth, the starch or the carbohydrate present in our mouth starts digestion at the mouth only by salivary amylase, and it is converted into a simpler sugar which is maltose sugar. So this is the function of salivary amylase in mouth. Next, the food goes into the food pipe, where the peristaltic movements push the foods down the entire alimentary canal. From food pipe, the food goes into the stomach. In the stomach, we can see two major things. One, stomach releases acid. This is the hydrochloric acid that stomach releases. The walls of the stomach release this acid so as to kill the germs whichever have entered the food particles along with the food we have eaten. And also it helps in softening of the food so that its digestion process is quite easy. Next is the mucus. As stomach is releasing acid, and acid is very corrosive, so stomach wall also releases mucus which protects the stomach lining itself from the acid produced by the stomach. In the stomach, the primary enzyme released is pepsin. The proteins that we eat start their digestion in the stomach. The proteins are converted into simpler peptones. Let me tell you that proteins are made up of small amino acids. Suppose that these are small amino acids and they form chain, or they can form three-dimensional structures or very complex structures to form proteins. So proteins are three-dimensional complex structures. When these are broken down into small chains or groups of amino acids, these are called peptones. So proteins are basically partially digested in stomach with the help of pepsin. Now the food moves into the small intestine. Now in small intestine many things happen. First of all, the liver produces bile juice which is stored in gallbladder. The bile juice from gallbladder and the liver, they also enter the small intestine, and there is a pancreas, another gland that also releases its digestive secretions called pancreatic juices inside the small intestine. So basically small intestine is releasing juices from gallbladder, from pancreas, and it has its own digestive enzymes. The function of bile is emulsification of fats. It breaks down the large fat globules into smaller droplets which can be easily digested. Now let us see some of the enzymes released by the small intestine and how the digestion takes place. The enzymes of small intestine contains maltase, which breaks down maltose into glucose and glucose. Lactase breaks down lactose into glucose and galactose. Sucrase breaks down sucrose into glucose and fructose. Trypsin breaks down the peptones, the partially digested proteins, into amino acids, and lipase digests the fats and lipids into fatty acid and glycerol. All these compounds are the simplest form of substances that can be absorbed by our blood and utilized by our body for various other functions. So in this manner we say that the digestion of food is completed in small intestine. The remaining undigested food goes into the large intestine, and the reabsorption of water takes place, and the remaining solid matter called feces is now expelled out of the body through anus. Apart from all this, small intestine contains small finger-like projections called villi which have blood supplies, and the entire digested food inside the small intestine is absorbed into this blood stream. This is how digestion process is completed in human beings. This is all about nutrition in plants and animals for today. Thank you very much, and for more details you can log into www.dnaeducation.com.